medicine monthly meetings. This month, we are honored to be hosting Dr. Amin Ardestani from Germany. He's Iranian, but of course he's in Germany, so he is regarded as an international instructor. It's our great pleasure and honor to host him today. The talk today is I will later say the title of the talk. Just let me give a brief introduction of Dr. Ardestani. He has completed his bachelor degree in biology from Tarbiyat Madaris University in Tehran, Iran. Later on, he completed his master's degree in biochemistry and biophysics from University of Tehran. And then in year 2013, he completed his PhD in biology from the University of Bremen, Germany. And after that, he is still in that university, and now he is a junior group leader at the Islet Biology Laboratory at the University of Bremen, Germany. And today he's going to speak for us about the inhibition of PHLPP1 and 2 Phosphatases Rescues Pancreatic Beta Cells in diabetes. Sorry, I'm not in this field, so it's a little bit difficult for me to pronounce those kind of expressions. Dear Dr. Ardestani, once again, I just wanted to thank you for accepting our invitation for the talk today. And these talks are designed so that our students get to know the international professors in the world and know what they're working on. And these are good means to start some communication and for some collaboration. And later on, for sure, we will more use your knowledge and expertise in our projects at the School of Advanced Technologies. We are 4 or 5 major fields in this school, and I think at least in 3 or 4 of them, they can use your help and knowledge. Once again, thank you, and we would be happy to have your talk today.
Okay, thank you very much. I just want to ask you, do you guys have my screen now?
Not yet. It says you have started the screen sharing, but I cannot see it yet.
I actually see it.
Now it's there?
I am able to see your screen. I'm not
everybody,
sure if Dr. Batuli is.
So everybody's able to see my screen, right?
Yeah, probably that's my problem. So please go on, Professor.
Okay. So
yes,
I'm very happy to
see.
Okay, I'm very happy to be with you here today, and I would like to thank my colleagues and at the School of Advanced Technology Medicine for their very kind invitation, especially Dr. Tabatabai, a Vice Dean of International Affairs, and any other organizers. And I would also thank the Dr. Azizi, our colleagues and collaborator at Department of Molecular Medicine. And in principle, our major research line is to understand the biology of insulin-producing pancreatic beta cell under physiological condition and also at diabetic state. Today, I'm going to present one of our recent project where we identify a strong regulator of beta cell turnover in diabetes. So let's begin with some introduction on the topic. As you know, the several organs in our body play a major role in blood glucose homeostasis by maintaining the balance between the uptake and storage of sugar. So blood glucose level rise after diet or in response to hepatic glucose production. So this will trigger production of insulin from pancreas, in particular islet of Langerhans. Insulin is a key factor for sugar to enter to peripheral tissue, muscle, and fat. As you can see, interaction of several tissues, several metabolic organs like a liver, intestine, pancreas, and muscles. So, and how they cross-communicate with each other, regulate energy homeostasis and glucose metabolism in the body. So in rest of my talk, I will focus on pancreas in general and in particular, islet of Langerhans. So, diabetes is a complex metabolic disorder when glucose homeostasis is compromised. So, diabetes is estimated to affect more than 400 million people worldwide. So, this number with the number of people with diabetes, so projected to reach more than 570 million by 2030. So within this population, approximately 90% of those affected will have type 2 diabetes. So the rest, which is 10%, will have type 1 diabetes and other form of rare diabetes. So despite available treatment for diabetes, which can control the disease and reduce diabetic complication, so far there is no cure for both type 1 and type 2 diabetes. So as I said, the pancreas is the very important metabolic organ in your body. So structurally, pancreas has two main compartments. So exocrine part, which include acinar cell and ductal structures. So which is responsible for production of digestive enzyme in our body. So exocrine part makes 98% to 99% of pancreas mass. Other part is endocrine pancreas, so which consists of the islet of Langerhans. Islet of Langerhans, as you may know, at least have 5 different cell types. So in particular, pancreatic beta cell and alpha cell, which is responsible for production and secretion of insulin and its counter-regulatory hormone glucagon respectively. So beta cell failure, or in other terms, loss of beta cell function and mass, is a common hallmark of both type 1 and type 2 diabetes. So as you can see in type 1 diabetes, the selective autoimmune attack destroy pancreatic beta cell, which leads to lack of insulin production and development of hyperglycemia. So in this case, the cytotoxic T cell play a major role for beta cell destructions. So on the other hand, type 2 diabetes is a complex multifactorial disorder, basically which is characterized by peripheral insulin resistance and as well as critical relative decline in the beta cell function and mass. So there are several diabetogenic stimuli, including glucotoxicity, lipotoxicity, and inflammation, which can trigger ER stress, oxidative stress, DNA damage response, which all collectively leads to impaired insulin secretion and beta cell apoptosis. So as you can see here, there is an inverse correlation between beta cell mass and fasting blood glucose. In this model, higher fasting blood glucose represent lower beta cell volume. So if you can see here, for example, in the type 1 diabetes, so with a higher fasting blood glucose level, you have a lower beta cell volume. So this is kind of true for also type 2 diabetes people, so and the people also with the impaired fasting glucose. So this indicates the importance of functional beta cell mass in regulation of glucose homeostasis and protection from diabetes. So programmed cell death or apoptosis is a hallmark of diminished pancreatic beta cell mass in both type 1 and type 2 diabetes. There is a negative correlation between beta cell apoptosis and beta cell volume or insulin positive area in type 1 diabetes and type 1 and type 2 diabetes. Here, this is a representative example from the people, the pancreas isolated from the type 1 diabetic people. As you can see, there is an absolute decline in the beta cell area, so that then you have the proportional increase in the number of the apoptotic beta cells. So this is true also for the people with type 2 diabetes. And also here you see that the beta cell volume, so in the obese people with the impaired fasting glucose or the established people, as the people with established type 2 diabetes. So also this is true in the lean people. If you compare with the healthy individual, also the beta cell volume is basically shrink almost to 40 to 50%. So, and then in the inverse correlation, and you can see the sharp increase in the number of apoptotic beta cell, which is evident in the both obese and the lean individuals. So it is also important to note here that the other mechanism, including, for example, the beta cell differentiation or failure of adaptive expansion due to impaired proliferation have emerged as a possible cause for this beta cell decline in diabetes. So, I mean, in addition to basically apoptotic beta cells, so the differentiated beta cell or the beta cell which is not able to proliferate, all collectively can also contribute in this sharp decline in beta cell mass in both type 1 and type 2 diabetes. So, as I presented, the loss of functional beta cell mass is a common pathologic element of all forms of diabetes. Thus, the major aim in the field is to identify therapeutic tools to rescue beta cells from apoptosis. In this case, the identification of cellular signaling pathway that regulates survival, proliferation, and regeneration of pancreatic beta cells, as well as their mechanism of action, is very important first for understanding the disease mechanism, and second for the discovery of new targets for beta cell targeted therapy in diabetes. So in our lab, in the Islet Biology lab, we are working over a decade on the one of important signaling pathway for the development, tissue homeostasis, and organ size, which is called the Hepa signaling pathway. So basically, we have two general status for Hepa pathway on and Hepa pathway off. When the Hepa pathway is on, the kinase MSD1 and 2 can phosphorylate the adapter protein here, called the SAV1, so, and also MOB1. So MSD1 in cooperation with the SAV1 and MOB1 is able to phosphorylate and activate the kinase LATS1 and 2. So then active LATS1 and 2 subsequently phosphorylate and inactivate transcriptional coactivator here, called the YAP and TAZ. So after phosphorylation, the YAP TAZ can basically sequester in cytoplasm and then lose their regulatory activity in terms of transcriptions. So cytoplasmic YAP and TAZ can go under further phosphorylation, which leads to eventual ubiquitin-dependent degradation by proteasomal machinery. So in contrast, so when the Hepa pathway is off, the MSD and LATS kinases present minimal activity, and YAP and TAZ can freely translocate to the nucleus. So they would be able to interact with the many transcription factors. For example, here in particular, the TIT family transcription factor, and induce their gene expression program, which is basically responsible for the anti-apoptotic response, regulation of cell cycle, and promotion of differentiation. So in our previous work, we were able to identify that MSD1 is a core kinase of Hepa signaling pathway, is a key regulator of beta cell survival and insulin secretion in diabetes. MSD activity, as represented by MSD phosphorylation, is highly upregulated under diabetic condition in both in vitro and in vivo condition. So we were able to block MSD1 genetically or chemically, and then we were able to restore beta cell viability and function and protect from diabetes in animal model of diabetes. So how does MSD1 work? So in summary, MSD1, so in cooperation with AKT, basically AKT is anti-apoptotic, is a very important anti-apoptotic protein in beta cells. So there is a mutual inhibition between MSD1 and AKT. So both in cooperation together can regulate JANG and also can activate the induction of BEAM. BEAM is the mitochondrial BH3 only proteins, so which can activate the intrinsic or mitochondrial pathway of cell death. So you have the classical cascade here, the activation, translocation, and activation of BAX or apoptotic BAX protein, and then inactivation of anti-apoptotic protein BCL2. So this leads to release of cytochrome C from mitochondria, formation of apoptosome, and then activation of the initiator caspase 9. So caspase 9 was able to activate caspase 3, and then so ultimately that can lead to the induction of apoptosis in beta cells. So importantly, caspase-3 also through the positive feedback loop can also cleave the MST1. So what happened after MST1 cleavage? The truncated form of the MST1 is able to further potentiate the apoptosis. So this is one distinct pathway how MST1 induced beta cell apoptosis. There is also other distinct pathway. The MST1 is able to interact and phosphorylate PDX1. PDX1 is a very important transcription factor in beta cell, which is responsible for beta cell development, insulin production, and anti-apoptotic response. So after phosphorylation, here the PDX1 is marked for the ubiquitin-dependent proteasomal degradation. So, and finally, ultimately, MST induced impaired insulin secretion through degradation of PDX1. So, at the time, the question remained to be answered: what is the regulator, upstream regulator of MST1? Or in particular, what is the common regulator of MST1 and then upstream AKT signal here? And we found a great candidate, the PHLPP phosphatase, in the short form, easy form, we call it Philip. Philip phosphatases as a common regulator of both MST1 and AKT has been identified before in the context of cancer. So as you can see here, the important Philip target is MST1. One of the important Philip target is MST1. MST1 is able to directly dephosphorylate the important inhibitory site in the MST1, which is called Tehran 387. So after that, so the other side, which is the opposite, is activatory, is a Tehran 183 become hyperphosphorylated, which leads to MST1 activation and also induction of apoptosis. The other Philip target is AKT with a similar mechanism but different output. Here the Philip1 is able to dephosphorylate AKT is a very important site in the hydrophobic region, which is serine 473. So upon dephosphorylation, this leads to the AKT deactivation and loss of the survival and growth signals. So the Philip or which is a Stanford for the pH domain leucine-rich repeat protein phosphatases. Those are enzymes that belongs to the PPT2C group of phosphatase, and so the family has two isozymes, the Philip1 and Philip2. So, and then you can see how much they're similar and how much are different. So also the Philip1 has 2S plus variant, also Philip1 version alpha and also beta. So in the rest of my talk, I'm going to focus on Philip1 beta because this is more established S plus variant of Philip, and also this is the predominantly expressed in the beta cells. So initial study identified Philip1 as a hydrophobic multiphosphatase which antagonizes pro-survival signaling pathway. It is well established that Philip1, when activated in response to cytotoxic stress, mediates cell death through the phosphorylation of multiple substrates. Here you can see as I said, the AKT is important substrate for Philip. So there is also a 6 kinase and also protein kinase C. In all the case, in these three cases, AKT, S6K, and PKC, the Philip dephosphorylation leads to the kinase inactivation. With the exception of MST1, as I said, the Philip dephosphorylation leads to MST1 activation. So all collectively can also participate in regulation of beta cell survival. So in the regulation of cell survival. And what makes Philip more relevant to diabetes is that the Philip1 single nucleotide polymorphism, or SNP, has been recently associated with the development of type 2D in some African populations. Also, in line with the genetic association, the Philip1 expression is elevated in adipose tissue and skeletal muscle of the human obese individual, including patients with type 2 diabetes, indicating an important role for Philip1 diabetes progression. So to check for Philip regulation beta cell, we expose the isolated human islet and then the rat insulinoma cell line, which is called INS1 beta cell, to diabetic milieu, which is here in principle is long-term elevated glucose concentration. As you can see, the Philip1 was highly upregulated in the INS1 beta cell and also in the isolated human islets. So both Philip1 and Philip2 showed a strong response when you compare to the untreated samples. Also, Philip levels were increased in islet isolated from diabetic, high-fat, high sucrose treated mice, as you can see here. So there's a strong upregulation of Philip1 and Philip2. Also, Philip levels were increased in islet isolated from diabetic. Another model of type 2 diabetic, the obese diabetic leptin receptor deficient DBDB mice. So in the which column they stand for DBDB mice. And in both model, diet-induced obesity and the genetic-induced obesity and diabetes, the Philip1 and Philip2 are strongly upregulated. Also, the beta cell specific expression of Philip2 was confirmed by double staining for Philip2 and insulin from pancreatic section from high-fat diet in comparison to the normal diet fat control mice. So this data show Philip1 is markedly elevated by pro-diabetic condition in vitro and in vivo. So to assess the role of Philip in beta cell death, we check whether Philip overexpression alone is sufficient to promote beta cell apoptosis. So here we use the adenoviral vector for the efficient overexpression of Philip1 in both INS1 cell and isolated human islets. So as you can see, the Philip overexpression induced beta cell apoptosis here as represented by induction of the Kili form of caspase-3. So you see the individually also when we combine the overexpression of Philip1 and Philip2, which we got the strongest response here. Also in the case of human islet, Philip overexpression induced here the apoptosis as represented by number of the tunnel positive beta cells. Also, overexpression of Philip impaired glucose-stimulated insulin secretion or GSIS in isolated human islet. What is the GSIS? GSIS in the simple informatic test, which can give you a lot of information about the beta cell functionality. So when you switch from the basal, which is basically is a low glucose, to the stimulated condition, which is the high glucose, you do see the strong response in the insulin secretion. But this is the laxy infected control human islet. But when you compare also the human islet, which is overexpressing Philip1 and Philip2, you see that also response is almost 50% decrease. So you see the data better on when we presented data as a stimulatory index, which is basically division of the stimulator to the basal, and then you see the statistically significant downregulation of the index when we overexpress Philip. So this suggests Philip has a detrimental role on both beta cell survival and insulin secretion. So to investigate whether Philip directly induced beta cell death in vivo, we use the polyplus transfection reagent, which is called JETPI. So what is JETPI? JETPI is the polyethylenamine-based delivery system for safe and efficient introduction of nucleic acid in the tissue. So the JETPI has to be incubated with the, let's say, your construct of interest. Here is we use the combination of Philip1 and Philip2, and for a control, we use the GFP expressing construct. So a solution of JETPI carrier complex with either HA labeled Philip1 and Philip2 or GFP control expression construct was injected IP into non-diabetic wild-type mice. So mice were given a total of 5 injections every alternate day and sacrificed 1 day after last injection. So a staining of pancreatic section using HA antibody shows the successful upregulation of Philip1 in the pancreatic islet. Also, the chronic administration of Philip significantly increased the beta cell apoptosis as compared to control GFP, again represented by the number of the tunnel-positive pancreatic beta cell. As I said, the MSD1 and AKT are two well-established targets for Philip-1. Also, here the Philip-1 expression. So the idea was to see what happened to those two different targets. Also, when we overexpress Philip in the pancreatic beta cell, Philip-1 overexpression, as you can see, reduce AKT-13 473 phosphorylation in INS1 cells. So in contrast, the Philip is able to increase the activatory side of the MSD1. So there is a basically reciprocal regulation between MSD1 and AKT when we overexpress the Philip in pancreatic beta cells. So in contrast, also Philip knockdown in INS1 cells enhance phospho-AKT level, as you can see. So when we block or when we knock down P1 individually or P2, or when we combine, when we knock down both together, you can see the strong upregulation of phosphorylated form of AKT1. So this is also consistent with our observation in Philip in isolated islet from Philip knockout mouse, so that you can see in the absence of Philip-1, the AKT phosphorylation is increased. So as the Philip substrate, AKT and MSD1 can mutually inhibit each other, several complementary experimental settings were designed in order to investigate the Philip AKT-MSD crosstalk. So we started with overexpression of mirror AKT. What is mirror AKT? Mirror AKT is a constitutively active form of AKT with a myristylation sequence which attached to the membrane. So mirror AKT is highly active and also is not sensitive to the Philip. So as you can see, overexpression of Philip so leads to activation of MSD1 and also cleavage of caspase-3 and caspase and PARP. So here you see the mirror AKT overexpression, block Philip-induced MSD1 activation and caspase-3 and PARP cleavage. So in the second experiment, INS1 cell were transfected with a phosphomimetic mutant of AKT1, in which serine 473, as we discussed, is permanently replaced with a phosphomimetic amino acid aspartic acid. Thus, AKT no longer be dephosphorylate by Philip and constitutively active. Also here, also overexpression of the mutant AKT suppress MSD1 activation and beta cell apoptosis triggered by Philip overexpression. So this indicates the critical role of AKT phosphorylation in regulation of AKT as well as MSD1 activity and subsequent beta cell survival. In third experiment, the kinase dead mutant of MSD1 overexpressed. What is the kinase dead mutant? The critical lysine. So in the ATP binding packet of MSD1 is mutated, so it cannot receive any ATP, and thus the kinase is entirely inactive. So mutant overexpression can antagonize the pro-apoptotic effect of Philip overexpression in beta cell, again as shown by reduced caspase-3 and PARP cleavage. So given that the Philip mediated the phosphorylation of MSD1 at Tehran 387 inhibitory site increased the activity of MSD1, we came with the idea to see whether such phosphorylation mediate the Philip-dependent regulation of MSD1 beta cells. INS1 cell again were transfected with a phosphomimetic mutant of MSD1 in which Tehran 387 is permanently replaced by the phosphomimetic amino acid glutamic acid. Thus, MSD1 is no longer dephosphorylate by Philip-1, and MSD mutant mimics the inhibitory phosphorylation of MSD1, which rendered the protein inactive. So in this case, the overexpression of MSD mutant reduced Philip-induced beta cell apoptosis, which shows against the important role of MSD1 dephosphorylation by Philip in the regulation of MSD1 activation and also induction of beta cell apoptosis. So to further support the role of MST1 as the main mediator of Philip-induced apoptosis in beta cell, we isolate islet from MST1 knockout or wild-type lethal mice. As you can see, the MST1 knockout islet were largely resistant to Philip-induced beta cell apoptosis. As I presented before, and the chronic exposure of beta cell as well as human islet to high glucose upregulate the Philip-1 levels. But the question remains to be answered, how does high glucose mechanistically induce Philip? First, we check the regulation of the Philip at transcriptome level when we treat them with the high glucose. As you can see in the both case of insulin beta cell and human islet, there was no change in the Philip-1 or Philip-2 mRNA level. Also, we look at the protein stability also on how fast the protein. So Philip-1 and Philip-2 in this case can be degraded by proteasomal machinery. Here we use the protein cyclohexamic chain protein assay. So to see how fast the Philip-1 or Philip-2 can degrade. Despite the higher induction of Philip-1 or Philip-2 at 0, the rate of degradation is not slower in high glucose treatment, even faster. So this excludes the possibility that Philip-1 or Philip-2 can be regulated at the post-translation level by high glucose. So the next level we are going to look at that is the Philip-1 or Philip-2 translations. So the mechanistic target of rapamycin complex 1, in short form mTORC1, is the master regulator of protein translation, which controls major cellular function, here in this case, such as metabolism, growth, and survival. And a high-profile study used high-resolution transcriptome scale ribosome profiling to show that subset of mRNA, which are specifically regulated by mTOR at translation level, consist of the established 5' terminal oligopyrimidine in the short form TOP, or previously unrecorded top-like motifs. So it means the mRNA, so mTORC1 through the classical pathway can regulate the mRNA which has the top or top-like motifs. So we performed the in silico analysis and identified that the 5' untranslated region of the Philip-1 mRNA contains a top-like motif. Also, Philip-2 mRNA contains top-like motif, proposing a potential mRNA-dependent regulation of Philip-1 at the translational level. So that was one piece of information. Other piece of information, we have previously reported sustained hyperactivation diabetic islets. So under diabetic milieu, the mTOR complex 1 is highly activated, which can block the other mTOR complex, which is basically the mitogenic complex, mTOR complex 2. So mTOR activation reduces the mTOR2 activity, which leads to beta cell death and impair function. So when we block the S681, a downstream target of mTORC1, we could rescue or restore the mTORC2 level, and also this restore beta cell survival and improve beta cell function. So because mTOR signal can induce Philip, we hypothesize that the expression of Philip-1 and Philip-2 under diabetic condition is upregulated at the translational level. And we have used a method which is called AHARA-RIBO to isolate the polyribosome in order to assess the active protein translation in the cells. The AHARA-RIBO is minimally invasive non-canonical amino acid tagging in which we isolate the active polyribosomes. The protocol relies on the pulse incubation of cell culture with a modified amino acid L-azidohomoalanine, which is called AHA. So then a small molecule like cyclohexamide block the nascent peptide attached to the ribosome. So then a biotin tag is linked to the newly synthesized AHA labeled protein. And finally, we are able to isolate the mRNA associated with polyribosome and quantify them by the qPCR. So as you can see, the high glucose treated insulin cells and islet isolated from long-term high-fat feeding diabetic mice display higher percentage of Philip mRNA bind to the polysome. So it means, so the Philip1 and Philip2 active translation is upregulated by high glucose. So in order to see whether mTOR regulates Philip expression, mTOR signaling was inhibited by a small molecule inhibitor rapamycin. So here you see the quick snapshot of mTOR1 signaling. So you have two arms. So in one arm you have the S6K1 kinase, and also you have the ribosomal protein S6K, which is directly phosphorylated by S6K1. So in other arm we have protein 4EPV1, which is a stand for eukaryotic translation initiation factor 4E binding protein 1. So basically the 4EPV1 is the blocker of the protein translation, and mTOR1 is the blocker of blocker, which stimulates the protein translation. So as you can see in both beta cell and human islet under glucotoxic condition, so as you can see, the mTOR inhibition by rapamycin can reduce the high glucose induced Philip1 and Philip2 upregulation. So, and as you can see, all the mTOR readout here, high phosphorylation of S6K1, phosphorylation of S6, and phosphorylation of 4EP1, which is highly induced by high glucose, is completely reversed by rapamycin. So we obtained a similar data also in the isolated human islets. Also, selective inhibition of endogenous mTOR by short, small interference RNA against the raptor, which is basically raptor is the essential component of mTOR1, and also the siRNA is targeting S6A1. So as you can see, again here in this case, high glucose induce a strong response in the Philip1 and Philip2, so which is accompanied by higher phosphorylation of MSD1 and activation of the caspase-3. So also the mTOR readout is highly up. So once we block the raptor to block mTOR signaling, the high glucose induced Philip1 and Philip2 upregulation is drastically reduced, which is basically, this is in line with the lower phosphorylation of MSD1 and complete termination of the mTOR readout. So also we obtained a similar data when we block mTOR signaling through inhibition of the mTOR downstream signal S6A1. So to further check whether mTOR or S6K1 signal is a principal regulator of Philip1 in beta cell, we overexpress the active form of S6K in insulin cells. So as you can see, the sustained S6A1 of overexpression effectively induced Philip1 and 2. This was combined with high phosphorylation of MSD1, and also you can see the phosphorylation of ribosomal protein S6K is highly up. So also the other method has been applied for the other loss of function method has been applied for the sustained activation of mTOR1. So in the second model of mTOR activation, we isolate MEF or mouse embryonic fibroblasts from wild type or TSC2 knockout mice. What is TSC2? TSC2 is a strong regulator of a strong inhibitor of the mTOR1. So in the absence of TSC2, the mTOR is sustainedly activated and also can activate its downstream signaling. So in the TSC2 knockout MEF, as an experimental model of the constitutive mTOR activation, so as you can see, the Philip1 and Philip2 was highly upregulated compared to wild-type MEF. And the blocking mTOR signaling by rapamycin resulted in a marked decrease of Philip1 and Philip2 protein expression in the TSC2 knockout cells, but not in the wild-type MEF cells. So in next chapter, we decided to check whether Philip1 or Philip2 upregulation is necessary for beta cell apoptosis. So what we did, we isolate the islet from wild type and the Philip knockout mice, and then expose them to the glycohypotoxic condition as well as mixture of pro-inflammatory cytokine interleukin-1 beta and interferon gamma. Here, the combination of the high glucose and free fatty acid palmitates is a condition which mimics type 2 diabetes. And then using the cytokines, which is basically the pro-inflammatory nature, is kind of the condition that mimic type 1 diabetes. So as you can see, the Philip deletion potently inhibits cytokine as well as high glucose palmitate induced beta cell death. Also, apoptosis triggered by cytokine as well as by mixture of high glucose palmitate was significantly abolished by knockdown of Philip1 or Philip2 individually or together in isolated human islets. As you can see, so this is the individual knockdown of Philip1 or Philip2. So in the purple column is when we block the Philip1 and Philip2 using the combination of siRNA against Philip1 and Philip2 in the single condition. So since Philip1 and Philip2 silencing improved beta cell survival under diabetic condition in vitro, we hypothesized that Philip1 deficiency might be beneficial against beta cell injury and diabetes development in vivo. To do so, we tested whether Philip1 knockout mice are protected from diabetes progression in multiple low-dose streptozotocin or STZ model of beta cell destruction and diabetes. What is streptozotocin or STZ? STZ is basically is a beta cell toxin. Is structurally is the sugar analog, which can potentially uptaken by cell expressing glucose transporter number 2. As beta cells are selectively expressing such transporter, the STZ is the major fraction of STZ can be uptaken by beta cells. Upon uptake, STZ is metabolized and which leads to induction of the oxidative stress, ER stress, and induction of DNA damage response, which can all collectively participate in the induction of beta cell apoptosis and destruction. So, as you can see here, so STZ induced the progressive hyperglycemia in the wild type mice. So first of all, at the non-diabetic condition, there is no change there is no change in terms of glycemia between wild type and Philip knock-on STZ. But you see the robust protection in the Philip knockout, which is injected by STZ. You do see the data better here when we quantify for area under curve. You see the very robust increase in the wild type STZ injected wild type mice, but in the direct comparison, you can see the powerful protection by Philip knockout. Also, when we look at the glucose tolerance data, so glucose tolerance data or GTT is a simple informatic metabolic test. So in this case, we inject sugar to the mice and we see how fast the body can clear the sugar from blood over the time. As you can see in the STZ injected wild type mice, you see the very robust impairment in the glucose tolerance. But however, also in contrast, the Philip knockout mice, which were injected with the STZ, show the very significant protection from impairment of GTT. Also, glucose-induced insulin secretion in vivo was fully blunted in STZ-treated wild-type mice, as you can see here. So in contrast, brief knockout animal presented a marked restoration in the insulin secretion 15 minutes after glucose challenge compared to the STZ-injected wild-type controls. So in line with the metabolic improvement, beta cell mass was significantly restored in Philip1 knockout compared to STZ wild-type mice. So to determine whether the regeneration of beta cell mass was a result of increased beta cell number due to enhanced beta cell proliferation, or rather is because of decreased beta cell apoptosis, we further assess the effect of Philip deletion on beta cell survival and proliferation. As you can see here, the data for the beta cell proliferations, we use the Ki-67 staining to label proliferating beta cells. As you can see, basically there is not much change between wild type and the P1 or PHLP1 knockout mice under non-diabetic condition, but there is the compensatory signal in response to STZ injury in the wild type STZ, which is almost double by PHLP knockout, STZ injected PHLP knockout mice. So it means the PHLP knockout is foster proliferation in the context of STZ. Also, when we look at the beta cell apoptosis, again here, which is labeled by tunnel staining, so you see the robust induction in the number of apoptotic beta cell in the STZ injected wild type mice, which is significantly corrected by PHLP knockout in the context of STZ. So here we look at the islet morphology at different cohorts. So here you see the typical islet structure of the rodent islet. You see the majority of cell insulin positive beta cells, which is placed in the core of islets. And then you see the minor fraction from the glucagon positive alpha cell, which are in the corner. So islet cell, as you can see, islet cell from STZ treated wild type mice were structurally disrupted with a fewer insulin positive beta cells and proportionally more glucagon positive alpha cell compared to untreated wild type mice. So in contrast, the percentage of glucagon positive alpha cells as well as the insulin positive beta cell in STZ injected PHLP knockout islet was similar to the non-STZ treated wild type control mice, which is basically confined to the rim of the islet. So here see the quantification of data. As you can see, you see almost the very strong induction of the alpha cell versus also very sharp decline in the number of insulin positive beta cells. So this islet architecture also, which is basically abnormal pattern, is corrected in PHLP knockout, STZ injected PHLP knockout islets. To further test the physiological relevance of our data in the second diabetic model, we check whether PHLP1 is necessary for long-term beta cell compensatory response in the diet-induced obesity mouse model of high-fat, high sucrose-induced diabetes. To do this, we place the wild-type PHLP knockout mice on either normal diet or high-fat diet for 17 weeks. In the case of high-fat diet, this leads to chronic hyperglycemia, insulin resistance, as well as beta cell failure in wild-type mice. So as you can see, after long-term high-fat diet feeding, wild-type mice develop the moderate hyperglycemia and also sharp impairment in glucose tolerance test, which was robustly corrected in high-fat diet treated wild-type knockout. High-fat diet treated PHLP1 knockout islets. And also you see the strong correction also in the glucose, GTD glucose impairment in the PHLP knockout mice which treated with high-fat diet. You see the data, also area under curve, which is basically extracted from GTT. And also here you see the insulin secretion data. As you can see, that PHLP deletion significantly enhanced insulin secretion in high-fat diet-fed mice. To further assess whether the improvement in glucose homeostasis in PHLP knockout mice were directly linked to the insulin secretion, we measured the GSIs ex vivo in isolated islet from high-fat diet-fed groups. Sorry, before that one, should not be here. So here. So this is the islet isolated for the ex vivo GSIs assay. As you can see, there is a very minimal response in the case of wild-type high-fat diet. So, but islet from PHLP knockout mice and the high-fat diet presented substantial increase in insulin secretion in response to glucose. We next ask a critical question: does reconstitution of PHLP reverse the enhancement in glucose responsiveness, which is evident in high-fat diet treated PHLP knockout islets? So you see here again, so an islet isolated from the different cohort of mice, you see minimal response in high-fat diet, wild-type high-fat diet. So, and then you see the substantial increase in insulin secretion in the PHLP knockout high-fat diet. So once we reconstitute PHLP1 through the adenoviral vector, we overexpress PHLP1 in the PHLP knockout islet, and you see the very strong response which we've seen in the PHLP knockout high-fat diet is completely reversed in the reconstituted islet. So this suggests that the existence of possible potential cell autonomous action of PHLP1 in the regulation of insulin secretion. So, similar to STT model, PHLP knockout mice displays a higher compensatory response in terms of beta cell mass relative to the wild type control mice. So, as you can see here, the very massive expansion of the islet isolated PHLP knockout mice under high-fat diet. So, this data suggests that the functional beta cell mass restoration as a key factor for metabolic benefit in the PHLP knockout mice. Also, like in line with the STZ data, so the proliferation rate is higher in the PHLP knockout mice, and also the number of the tunnel apoptotic beta cell is significantly decreased in the PHLP knockout high-fat diet treated mice. So because mTORC induced PHLP, as I presented before, we also investigated whether diabetes-induced PHLP expression is regulated by mTORC1 in the normal or high-fat diet treated mice. So similar to INS1 cell and human islet under high glucose, you see the very strong response in PHLP1, PHLP2, and phosphorylation of S6F1 and other mTORC targets, and also phosphorylation of MSD1. But once we block the mTORC signaling by rapamycin, so high-fat diet induced PHLP1 and PHLP2 is drastically reduced, which is in a strong correlation with reduction of the mTORC1 readout. So we obtained similar data in the other condition, in other islet, which is isolated from the parallel cohort. So, and then you see on the right panel also the quantification of data. You see the strong response to PHLP1 and PHLP2 in the high-fat diet, also which is significantly reduced by using the rapamycin to block mTORC1 signaling. So this is also true for phosphorylation of MSD1. So here it means there is a very powerful correlation between PHLP1, PHLP2, and phosphorylation of MSD1 in the context of high-fat diabetic islets. So to see whether observed PHLP1 or PHLP2 upregulation in animal model of diabetes is relevant to a human diabetes, we investigated islet from patients with type 2 diabetes. So you see the Western blot analysis of human islet from type 2 diabetic patients. You see the profound response in the PHLP1 or PHLP2 in islet isolated from the type 2 diabetic patients. So, and then also you see also here quantification, which shows the significant upregulation of PHLP1 and PHLP2 in the islet isolate from human diabetes. Also, a staining confirmed increased protein expression of PHLP2 in insulin positive area in the islet from patients with type 2 diabetes in comparison to the non-diabetic control. And then you see the very beta cell specific induction of PHLP2. So in order to understand the pathophysiological impact of increased PHLP1 in human islet in the context of type 2 diabetes, human type 2 diabetes, so we use the siRNA to genetically inhibit PHLP1 or PHLP2. First, as you can see, the genetic inhibition of PHLP1 or PHLP2 does not have any impact on insulin secretion in the control human islet. While genetic inhibition of PHLP1 or PHLP2 alone or together had significant improvement in terms of insulin secretion in the islet isolated from type 2 diabetic human islet. And also here you see in the terms of survival, beta cell viability was restored by genetic inhibition of PHLP2 in type 2 diabetic islet. So finally, this is our working model, how PHLP works. And as you can see here, also under diabetic condition, basically in particular chronically elevated glucose. So we have the sustained activation of mTORC. The question still remains to be answered. What is the mechanism or upstream regulator which connect the high glucose to mTORC? So that we don't know. But at the downstream level, mTORC can promote PHLP1 or PHLP2 translation by two different arms. So activation of S6A1 or blocking of 4-EBP1 proteins. So once PHLP1 or PHLP2 is up, so they can simultaneously regulate their downstream target AKT1 and MST1. So we have the phosphorylation inhibition of AKT1 and the phosphorylation activation of MST1. So which ultimately can induce beta cell apoptosis and impair beta cell function. So this is our summary. PHLP is upregulated in pancreatic section from patients with diabetes, also in the rodent model of diabetes, and also at the in vitro condition when we expose the beta cell line or human islets. So PHLP overexpression can directly impair beta cell survival and function. So PHLP negatively can regulate the AKT, anti-apoptotic AKT, and also induce pro-apoptotic MST1. So we identify mTORC1, sustained activation of mTORC1, so which links the high glucose to induction of PHLP1 at the translational level. So also genetic PHLP inhibition restore beta cell survival insulin secretion. So this has been shown in the isolated human islet in vitro, also or in the two animal model of type 1 and type 2 diabetes. So in the end, thank you very much for your attention. And I would like also to thank you, my colleagues and my students in the Islet Biology Lab in the University of Bremen. Also, I'm thankful of all the funding agency and also our collaborator, which is specifically provide the isolated human islets and also PHLP knockout mice to our labs. So now I'm happy to answer any question.
Thank you very much, Dr. Ardestani, for the really fascinating talk. I'm not in this field, but I could understand something. That's because of your nice talk. Thank you very much again for that. And I hope this would be a starting point for further collaborations in the future with you and with your lab, of course. For the questions, I just wanted to ask Dr. Azizi to help us because she is more expert in the field. So if she can manage the questions, we would appreciate.
I think you need to turn on the microphone, yeah?
Yeah, I did. Yes, she's here.
Okay. And Dr. Azizi, can you hear us? So, because I'm not able to hear her, I mean.
Yeah, please go on, Dr. Azizi.
Yeah, thank you. Sorry, unfortunately, I'm reading from the transition from the universe, so I cannot really access to the microphone. So this is the reason that I'm using just a passive microphone right now. Thank you, Dr. Ardestani, for your very nice presentation and very useful one. That was really interesting for myself. First, I'm asking the students that they are participating in this journal club, and if they have any questions, please go. It's me. Hello? Yes.
Dr. Azizi, I think you should go on. Probably students will ask their questions later.
Yeah, for sure. Dr. Ardestani, as he said, decrease or blocking the PHLP of FILIP1 or FILIP2 can help restore the beta cell mass. Yeah. So, what's your next step for treating? It's possible that you use some chemical compounds for somehow with as a drug for treatment of type 1 diabetes or type 2 diabetes. People, did you is it your next part for this subject?
Actually, what I understood from your question, because the quality is not really good, but you're asking me also what would be the next step also in order to like Philip or move the project to the translational level. So basically, yes, there are the current compound in the market which can block FILIP1 and FILIP2. And just to be honest, also we tested them also a couple of years ago. But the problem with the FILIP inhibitors, so the current FILIP inhibitor we have in the market is, there are two issues with them. One is the lack of specificity. So we got a lot of off-target. That means, so in addition to blocking FILIP1 or FILIP2, also they can block the other phosphatase. So we have the case of the off-target also, lack of selectivity in this case. The other issue was at least one of the inhibitor we use was interacting with the albumin in the cell culture media. So just to minimize that one, we had to increase the dosage of the compound to basically to the very late micromolar range, something like a 50 macromolar or 100 macromolar or 200 macromolar. So as you know that this is very much can increase the toxicity of the cells, and this can preclude the moving the compound to the clinical mouse model of diabetes also. So in the light of these two issues, so having the off-target effect and also having a problem with the interaction with the albumin, so we decided not to move on with the FILIP inhibitors. So, and then we decided to move for the alternative strategy. Now we are currently working with the company in the US, so which we apply the ASO technology or antisense oligonucleotide to specifically inhibit FILIP1 or FILIP2 in the our target cell, which is the pancreatic beta cells. So we don't have that much data at the moment, but we have the ASO. We further characterize the ASO, and the ASO were able to specifically inhibit or reduce the FILIP1 or FILIP2 level in pancreatic beta cell specifically in the rodent models. So now we are working on the basically dosage and also how often we have to treat the mice. So, and then once we find out all the little information about this technique, we would like to move on with the mouse model of diabetes. So, but in short form also that, okay, so I don't know, maybe in the future there will be more effective or more specific compound, but at the moment we don't use the current chemical inhibitor against FILIP1 or FILIP2.
Also great, because it's really interesting for myself as a person that is working in this which is field. And thank you for your answer. And the second question that I had that was about this FILIP knockout mice that you produced. How long did you check them in the animal facility? How long they could live? And did you check for any unusual growth in the islets of them after some time? For example, as an endocrine pancreatic somehow producing cells.
Yeah, I think it's a great question, and also of course we observe the mice. So first of all, let's say also we use them for the two different mouse model. One was STD, which basically we use pretty young mice, is 2-3 months old mice. But in the case of high fat diet mice, because of the length of the nature of the experiment, so we were able to keep them up to 7-8 months old. And also there was the other case, we kept the mice up to 1 year. So usually we didn't see, okay, we didn't look at any other tissues, but at least in the context of pancreas, we didn't see any abnormality or let's say the unexplained growth or something. But that's the good direction you are asking your question, because the PHLP1 or PHLP2, I mean, so in addition to be a pro-apoptotic mediator, those are the gene, basically those are the tumor suppressor genes, which restrict the replication and also differentiation in your target cells. So it has been reported that in the PHLP knockout mice, in a very old mice, almost 1.5 years and 2 years, also there was the case of developing the tumors.
Okay.
So.
And I can remember that in 2014, you published a very novel paper in Nature Medicine that talked about MST1. And after that, you were working about the chemical compounds to somehow regulate it in the diabetes people. What's your idea of which one of these two targets can be the one that you suggest, PHLP1 or MST1, as a target for maybe helping the diabetes people?
I don't know. It would be hard for me to choose between MST1 and PHLP. I mean, but to be honest, okay, there are some advantage with MST1. So now we currently have a couple of inhibitors in the market. So this one advantage. The other advantage, we almost finished most of the preclinical mouse models. Also, we have a lot of information how does MST1 works basically in a mouse. So moving through the translational phase, also, of course, I would choose MST1 inhibitor. But for PHLP, we are still at the earliest stage. Also, we just recently published, because most of the data I presented now. So we just recently published all the basic on also all the data what we have also in the mouse model of diabetes. So just to moving forward, we have to identify very low risk and highly efficient way to block PHLP1 in target cells. Also, this is the same disadvantage we have also with the MST1. So MST1 as well as PHLP1 and PHLP2 are tumor suppressor genes. We have to be very careful. So we cannot go for the permanent inhibition. So that increase the risk of the tumorigenicity in the body. So we have to think about also to go for very, very transient and also very effective strategy to block MST1 or PHLP or any other target in the pancreatic beta cell.
Thank you, Dr. Ardestani. Your presentation, that was really brilliant. I really enjoyed about it.
Thank you very much. And then I'm happy also to answer any other question if there isn't time.
Yeah, for sure. Dr. Zawatawi or Dr. Khalrazi, if you want to add any comment, please let me know that I can activate your microphone, or any of the other attendees. We would be happy if you have any questions. Okay, it seems everything was obvious to everybody, so there's no more question. Dr. Ardestani, I wanted to once again thank you for accepting our invitation to attend this meeting, and it was a really big honor and pleasure for us to host you. And as I said, we're not looking just for this meeting, and we hope our collaboration continues in the future, and we definitely need your knowledge and expertise and help, and like any of the other international instructors that we're approaching. Oh, there is one question. Let me see. Yeah.
Good afternoon, Dr. Ardestani and all the other participants, and Dr. Azizi. That was quite a brilliant presentation and quite a rich, informative research work. I actually really admire what you have done so far. My question is, looking at the molecular mechanism between mTOR and the PHLP1, when you people knocked down the PHLP1, was there any like some maybe compensatory mechanism from the PHLP1 as regards to maybe its effects on some other parts of the cell that could have maybe caused, for example, the initial inhibitory drug that people produce not to really, work properly. I don't know if you got my question completely.
If I understand you correctly, you're asking me if once we knock out PHLP1, is there any compensatory response or through the positive or negative feedback loop to the upper signal? Okay, in this case, I mean, in generally, I made the story very, simple. Because sometimes too much complication, we don't understand the model. So because for our own basically advantage, we always try to make the things simple, basically, which is not true in the very complex system that we are working, even in the cell culture system. So, but basically I present the data when you have a signal from complex 1. Basically complex 2 is responsible for activation of AKT, which is responsive to the insulin or IGF signaling pathway. But usually mTORC1, once is very highly activated, can send a message through the feedback mechanism to terminate the complex 2. So, and then there are many links here. I don't know if probably you know, but one of them is basically is complex 1 to the S6K1, and then is going to be the phosphorylation of the IRS protein. Insulin receptor substrate, and then which finally ultimately can inhibit the complex 2 and AKT. But in this case, we identify very novel loops, basically complex 1 to PHLP1 or 2, and PHLP1 or 2 are direct phosphatase for AKT1 and 2. So we think that one novel, at least in the context of beta cell, from signal from complex 1 to PHLP and PHLP to AKT. So in this case, with activation of mTORC or activation of PHLP1 or PHLP2, you can terminate the, let's say, anti-apoptotic or mitogenic signal, which is basically governed by the PI3K AKT1 signaling pathway. So, but this is very complex also, but but in this case, we only focus what happened downstream of the mTORC1.
Thank you very much, Doc. That was quite a brilliant explanation. I'm really happy, at least I asked that because I wanted to look at the immunological aspects of the pathway, and I'm really happy you could explain this. My second question is concerning the anti-PHLP1 and 2 drugs that you people produced. You said it had some complications and you had to increase the dosage, but again, I realized that this was kind of, lethal to the cells. Was there, could you really decipher, could you come out with the reason why the anti-PHLP1 and 2 drug didn't really actually work?
Actually, this is usually what we do basically when we want to test any drug also. Of course, it depends. Sometimes drug has, let's say, you have the really numerous publication before, and then you can have an idea or what to use, how to use, in terms of dosage or time or anything. So, but in this case, there was a few publications, which were reported also the high dose. But what we did in the lab when we got the compounds, and then we tried to do the titration also. We went from low dose to, let's say, high dose. High dose at the time for us was 10 or 20 micromolar. This is usually normal. So, because most effective dose for the drug is going for the late nanomolar. So, we reached from the late nanomolar to middle micromolar, and then we saw nothing basically in terms of AKT activation. Later, we realized from our collaborator that one of the compounds can really interact with the albumin. So in this case, you have two options. either you have to completely remove serum from your culture, which is not possible basically, because having serum in the culture is very essential for the maintaining the islet or cell line in the culture. So we choose the other strategy. We kept the albumin and we kept serum and then increased the dosage. But dosage was 50, 100, 150, 200 macromolar, which is very high. So we saw the AKT activation. It means the inhibitor works. But so in addition to that, we saw the very high toxicity. So basically the amount of compound we used that completely changed the color of media. So which was very unspecific to me also. The other issue, as I said, we did a kind of the pilot profile study against the PHLP1 and any other phosphatases. And then we were kind of surprised that PHLP1 in addition to, sorry, the compound in addition to PHLP1 or PHLP2 can block other phosphatases. So which is sometimes have a different outcome when you block. So in this case, we decided not to move on. Basically, the idea was, okay, to characterize the compound in the cell culture system. So confirm their activity, confirm their specificity, and then later on move to the very expensive, let's say, model like using them, also injecting to the mouse models, also in the different thing, also in the case of human islet transplanted to the mice. So, and then of course, in that time also we decided to also be completely off with the idea. So, and then we moved on, or we went on with the other tools, which is very exciting in this case. So as I said, we use the ASO or antisense oligonucleotides, which is basically labeled with a part of GLP-1. GLP-1, as you know, is an incretin hormones, so which is basically produced by gut and can act on the beta cells because there is a selective expression of the GLP-1 receptor and beta cells. What we did also in the collaboration with the Ionis company in the US, so we use the antisense against PHLP1 or PHLP2, which is linked to the part of GLP-1. So in this case, in theory, also this ASO can be 100% uptaken by the beta cells because the beta cell is the major source of GLP-1 receptor in the body. So we did, and then we got a very exciting data in the cell line, also in the mouse. So now at the moment, we are trying to find the optimum concentration for ASO and also the length of experiments. Once we finalize those information, we would like to move on with the preclinical mouse model of diabetes. Hello? Hello?
Yep. Yeah, Professor, we can.
Sorry, I lost you. I don't know.
Yeah, we can hear you, but just Aaron muted his microphone.
Oh, okay.
Probably he has no. Yeah.
I wasn't sure.
Thank you.
Able to hear my answer. Yeah.
yeah. He did. We all heard it. Thank you very much again, and I'm waiting for just 1 minute to see if any more questions coming up. If not, we will finalize this session. So I should thank again Dr. Ardestani for giving us this opportunity to host you. And also I do appreciate the time and attention of the attendees of this meeting. And we hope we have further collaborations in the future again.
Sure,
thank you very much again.
Everybody, I mean, all the participants, also I'm also thanking against the organizers, also you, Dr. Aziz and Dr. Tabatabai. And of course, I'm very open also. We already have the collaboration with the Department of Molecular Medicine, also with the Dr. Azizi. And then I would be also happy to extend our collaboration also to the other departments and also other labs also. And also I'm thanking also very great idea, also Journal Club also. So Dr. Azizi told me a bit late, but anyway, I'm very happy also to attend in such amazing clubs.
Yeah, thank you very much. By the way, you were busy, but accepted our invitation. We thank you again, and we just finalize this meeting now, and hopefully we can have attendees for the next month meeting as well.
Okay, thank you very much.
Thank you very much. Thank you.
Have a nice day, everybody. Bye.
Thank you. Bye.